Yes, so in my presentation, I will first focus on the great leap in technology that allows us today to use a genomic analysis to evaluate cancer risk from environmental factors. So what we've seen is that the sequencing technology now allows us greater power to identify all the mutations that are present in a cancer tumor, so in a cancer sample, and this allows us to really untangle all the different sources of mutations that happen there, and some of which are from environmental factors and some of which are not. So in the presentation, I will also go a little bit through how we can untangle these different sources of mutations. And then I will give some slides also on recent studies that aimed at validating these findings. So actually reproducing this genetic damage that these different type of environmental factors can do, and these have been reproduced mostly in cell lines, but also in some mouse studies. And finally, I just want to highlight some important ideas, like for example that this damage that come from that we see in the genomes that come from different environmental factors can be seen in also normal tissue, not just cancer tissue. So the cells are able to actually endure some of this damage quite well, as it is always a matter of a balance between the damage and the capacity of repair of them. So I will also show what happens in a particular patient that a case study that we published where a patient that had a particular DNA repair defect actually had a much more severe damage and developed a tumor because of that.